All right, we are looking at part 3. So part 3 talks about homeostasis and the transportation of substances across membranes. So we're looking at passive transport first. So passive transport first is basically the movement of molecules in the path of least resistance. So diffusion is the simplest form of passive transport. It works by kinetic energy of molecules. It's movement of molecules from an area of high concentration to low concentration. So that is, of course, what we're looking at for a passive transport. At the area or path of least resistance. Concentration gradient is the difference between the concentration of molecules across the space. Equilibrium, of course, is the concentration of molecules that is the same throughout space. So our goal is equilibrium in a transport. Diffusion happens across membranes. So this is the ability of a molecule to diffuse across the membrane depending on the size and type of molecule and chemical nature of that membrane. So things can dissolve in lipids, will go across the membrane because it is made of lipids. Osmosis is the movement of water from an area of high concentration to low concentration. This is also a type of passive transport. So the direction of osmosis depends on the concentration of the solutes on both sides of those membranes. So when we're talking about this, we're going to need to know the prefixes. Otherwise, we're not going to understand what we're talking about. So hypo means below, hyper means above, and iso is equal. So if you ever hear something called iso, that means it is equal or the same. So or equilibrium or homeostasis. The concentration of several molecules outside of the cell is lower than the cystosol in the solution of a hypotonic to the cystosol. So that's the example. So in this situation, water will diffuse into the cell until equilibrium is established. If it is hypertonic, that means water will diffuse out of the cell until equilibrium is established. If it is isotonic, there will not be any movement of water because the diffusion will go in and out at the same rate. You will have osmosis, but it is already at equilibrium, so there is no need for an influx or outflux of water from the cell. It will go gradually. Many cells function in a hypertonic environment, like freshwater organisms. So that means you have a lower concentration because of the cells constantly take on extra water through osmosis. You have single-celled organisms handle this by having a contractile vacuole. So that means it will an organelle that removes excess water. So basically stores it or expels it. The special vacuole uses energy to maintain equilibrium. Multicellular organisms respond to hypotonic environments by pumping solutes out of the cystosol. So plant cells also live in hypotonic environments. Typically, their roots are surrounded by water, which causes the water to move into the plant through osmosis. So if they're in a hypotonic state, that means they are allowed to take on more water. So this condition is called plasmolysis, a condition in which turgor pressure is lost. So the plant causes it to wilt. So some cells cannot compensate for the change, and this will cause it to burst. So you will have this burst is called cytosis. So facilitated diffusion. This is a type of passive transport where the molecules cannot diffuse rapidly. So across the membrane, even though there is a concentration of gradient. So such molecules may not be soluble. They may not be soluble through lipids. They're too large to pass through the pores of the membrane. This means that to get these molecules across, carrier proteins have to be used. Carrier proteins are move molecules from areas of high concentration to low concentration. So if they're too big, they have to have these carrier proteins. This does not require energy. Carrier molecules are specific to certain molecules. So diffusion through ion channels. So you would learn this in chemistry. Ions cannot diffuse across a membrane because they are not soluble in lipids. Ions channel through open pores in the membrane, and ions pass. So they are small enough to be able to pass through these gates. Some are always opens, other have gates. So these gates will open and close through electrical symbols when the stimuli is needed from external environment. So these stimuli control the ability of ions to pass through the membrane. All right, that is passive transport. Now we're moving on to active transport. Active transport of molecules against the concentration of gradient that requires energy. So cell membrane pumps. These pumps actively transport because they require energy. So a particular substance binds to the pump, which causes the pump to change shape and shield the molecule from the hydrophobic interior of the lipid bilayer. In other words, this will cause it to activate. It works the same as a carrier protein in facilitated diffusion. Makes sense. So it comes on there, you're able to have some active transport. Sodium potassium pump is an example of an active transport in animal cells that uses a carrier protein called the sodium potassium pump. So it functions moving both sodium and potassium ions up their concentrated gradients. Steps of the sodium, you have step 1, which is 3 ions bind to the carrier protein. At the same time, the carrier molecule splits a phosphate group of molecule of ATP. So this is your energy. The phosphate is bound to the carrier protein. The binding of the phosphate group changes the shape of the carrier protein, allowing 3 sodium ion molecules, to release into the environment. 2 protein ions located outside the cell bind to a carrier protein. This is step 4. 5. This causes the phosphate group to be released, thus changing the shape of the carrier molecule back to its original shape. So now this way we do not have too much sodium coming in. The 2 potassium molecules are then released into the cytosol of the cell, and the cell cycle is ready to repeat again. So if there's a need for your sodium, it will once again activate. This process creates an electrical gradient across the membrane, becomes positively charged outside the membrane and negatively charged inside, because you are losing those potassium molecules once they get in, or potassium ions. So endocytosis and exocytosis. This is part 2 of our notes. So some molecules are too big to be transported through facilitated diffusion or by a pump. To get these large molecules across, like food particles or protein, the cell uses exocytosis and endocytosis. So endocytosis is a process in which a cell ingests external fluid, macromolecules, large parts particles, and other cells. So the surface proteins can bind on the membrane, cause the membrane to fold up upon itself and form a pouch. Then it pinches off, forms a membrane-bound organelle called a vesicle, enclosing material that is brought into the cell. There are 2 types of endocytosis. It determines on what is brought into. So pineocytosis involves the transport of solutes and fluids, and phagocytosis involves the movement of large particles or other cells. So cells use this in their bodies to fight bacteria and viruses. These type of cells are known as phagocytes and allow lysosomes to fuse with the vesicle that contains this bacteria or virus, releasing digestive enzymes to try to destroy them. Exocytosis, this is the reversal of endocytosis. In this, proteins out on the outside of the vesicle fuse with proteins on the inside of the cell membrane, release their contents into the outside. So the Golgi apparatus packages the proteins into the vesicles, which they release to the outside of the cell. So basically, the Golgi apparatus will combine or packages these proteins into the vesicles and releases it out in order to get rid of this waste, or in sometimes to make sure that there is nothing harmful inside the cell. That is part 3. So you will need to complete the quiz. 7 out of 10, as always. Once you have done that, you are free to move on to part 4.